Hello and good morning to fellow friends and Dr. Suganti. Today we are going to talk about shotgun sequencing by us Group 7. Alright, our group consists 8 members, which is me, Nur Awali Midot Zarin, and Ami Zaim are the one editor for this slide and video. Ami is the one who making video, and while I am doing this slide and the introduction and the conclusion of this. The other members like Hasina Zara will talk about the history part, and the application part will talk by Mila. The advantage will talk by Hashira Mainis, while the disadvantage will talk by Nadira. For the discussion part, we have two things to talk about, which is case study and article. Case study will briefly talk by Ain, while the article part talk by Aini. And so that is all. So let's begin now.
Hi and Assalamualaikum. My name is Hasina Zahran binti Hasan. I'm going to present for the history part. Shotgun sequencing is a technique for determining the DNA sequence of an organism's genome in a laboratory setting. The approach entails randomly slicing the genome into a little DNA fragments, each of which is sequenced separately. In the 1980s, Frederick Sanger and his colleagues first utilized shotgun sequencing to sequence tiny genomes like those of viruses and bacteria. It gets its name from the shotgun's rapidly expanding, quasi-random short grouping. Shotgun sequencing originally comes from the idea that a huge sequence is split down into many smaller parts, almost like a shotgun shell does when fired. It breaks down DNA into many short segments at random, which are then sequenced using the chain termination method to generate reads. Performing repeated rounds of this fragmentation and sequencing yields multiple overlapping readings for the target DNA. The overlapping ends of individual reads are then used by computer system to combine them into a continuous sequence. That's all from me. Thank you.
Okay, next I want to talk about application in Sanger sequencing. Sanger sequencing, also known as a chain termination sequencing or detection sequencing. The process is based on the detection of labeled chain termination nucleotides that are incorporated by a DNA polymerase during the replication of a template. Okay, so we will refer in this table. They show the application of a shotgun sequencing. Okay, we will look the DNA sequencing and RNA sequencing in this table. The process is based on the detection of labeled chain termination nucleotides incorporated by a DNA polymerase during the replication of a template.
Assalamualaikum. My name is Hashira Mainis binti Hasan. Metric number E21B0484. I would like to present about advantage of shotgun sequencing. First, advantage of shotgun sequencing is whole genome shotgun sequencing is substantial faster than clone by clone sequencing since the mapping process ASCII. Second, whole genome shotgun sequencing required a fraction of the DNA required by clone sequencing. Lastly, when there is an exciting reference sequence, whole genome shotgun sequencing is very efficient. It's not much easy to put together a genome sequence, but matching it to a no reference genome. Shotgun sequencing is quicker and cheaper than approaches that required a genetic map.
Assalamualaikum and hi everyone. My name is Nur Nadira binti Azman. Today I want to present about shotgun sequencing. My part in this lab report is disadvantages. Okay, for the first point, disadvantages in shotgun sequencing is can introduce error in the assembly process. Second point is reconnaissance of different genomes. And last point in shotgun sequencing in disadvantages is may not be able to assemble detective sequences. This all for me.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nukrota binti Muhammad Rosli. Today I want to talk about the case study that involving in the shotgun sequencing that related to these natural resources. For the first point, in the case study, that is a shotgun sequencing was used on the 8 species in the Chrysobalanaceae family tree. When they use this method, it can help to detect the large fragment of DNA and the potential to get the phylogeny reconstruction. Okay, for the second point, in the case study that use a shotgun sequencing, that is the Chrysobalanaceae family tree, has not long been neglected in the molecular phylogeny, but it is rich in secondary chemical compounds that found in the family tree. Last point for the case study, that is the plastic genome, ptDNA, and mitochondrial sequences, mtDNA, was used in this case study should not be mistaken for definitive. That is because they ignore some of lineage and DNA information of the Chrysobalanaceae family tree. That's all from me. Thank you.
Assalamualaikum, my name is Nur Ali binti Muhammad Syedi, matric number E21A0570. I would like to discuss article title Environmental Shotgun Sequencing, its potential and challenge for studying the hidden microbial world. In this article, I focus on one very promising field on metagenomics, namely the use of the genome method shotgun for the random switching of DNA fragment for microbial in environmental sample. Switching a level of random environmental shotgun, first used just a few years ago and now used to test every imaginable microbe system from the human gut to water sludge residues, and have the potential to reveal and base observation into the hidden microbial world. One of the most important two conceptually simple steps in studying an ecosystem involves cataloging the type of organism and the number of each type. For a long time, typing and counting like that was an almost insurmountable problem in microbiology. This is largely because physical appearance does not provide a valid taxonomic picture in microbes. Appearance developed so rapidly that two closely related taxa may look very different, and two distantly related taxa may look familiar. This annoying problem was solved in part in the 1918 through the use of rRNA PCR. This method allowed the microorganism in the sample to be typed and counted phylogenetically based on the rRNA sequence. This method allowed the microorganism in the sample of the phylogenetically typed type and calculate based on the squinch and of their RNA genes found in all cell-based organisms. So we can fully realize the potential of the ESS to type and count species, including creating an automated yet accurate phylogenetic tree of each gene, determining which gene is most useful for which taxa, combining data from different genes, even if we don't know if it come from the same organism. Build a database of gene other than rRNA and compensate for the lack of sampling data. If these challenges are met, the ESS has the potential to rewrite many of the things we think we know about the phylogenetic diversity of microbial life. That's all for me. Thank you.
Alright, that is wrap. Thank you for listening our group members present about the shotgun sequencing. Hope you enjoy and learn well about the shotgun sequencing. And see you soon. Goodbye. Thank you.